So first of all, thank you very much to the organizer of this meeting for the invitation to talk today. It's great to see you in live, but I would also like to welcome all the people connected in Zoom. Today I will present a part of my PhD project that was conducted at the Swiss TPH in Basel and that I concluded last year. And especially this project was conducted in close collaboration with the Yale University. And I'm going to talk about the dispersal dynamics of the Asian tiger mosquito across the Alps. So I believe within this audience, I don't have to spend much word on introducing the real protagonist of this talk, so the Asian tiger mosquito, Aedes albopictus, one of the most invasive species worldwide that originate from Southeast Asia. And it's a diurnal urban mosquito of vector of different arboviruses such as chikungunya, dengue, and Zika. Here I report one of the latest distribution map from ECDC of Aedes albopictus in Europe. And we know after the first introduction in the late 70s in Albania and after 10 years in 2 sites in Italy, this mosquito was able to spread quite dramatically in all the Mediterranean basin. And going to the north, it reached its latitude as far as in the Netherlands. But what we have to keep in mind that Europe is crossed by the Alps. And the Alps are a biogeographical barriers for many organisms. So the question is how much they limit the spread northwards. Because there have been many models developed to try to understand in which area Aedes albopictus will be able to establish population. But how did it manage to spread so dramatically? We know that the active dispersal is limited, or this is a general assumption. And in another part of my PhD, which I'm not going to present today, I tackle the active dispersal of Aedes albopictus. And here I report the manuscript in case you're not familiar with it, and I encourage you to read it. This was a mark release recapture study conducted in southern Switzerland, where I found out that Aedes albopictus is able to cover larger distance than previously assumed, so about 800 meters. But we know that the key driver for this mosquito is the passive dispersal, which can occur in 2 life stages of the mosquito life cycle, so as eggs, which are passively distributed, especially across continent, and more at a regional level, mosquitoes are hitchhiking, and they are then displaced across region. So the general aim of this project was to disentangle the dispersal pathway of Aedes albopictus across the Alps in order to know its invasion history, to apply more targeted control and surveillance. Especially, I was particularly interested to know which are the origin and the introduction pathway across the Alps, and if the Alps are somehow a barrier for establishment of Aedes albopictus population. And also all the population that we find north of the Alps, are these really established or they are reintroduced every year through traffic routes, for example. So to do so, I used genetics, especially genetic markers as SNPs. These are single nucleotide polymorphism, so changes in one base of a DNA sequence. And to obtain SNPs, I used a next generation sequencing approach, namely double digested restriction site associated DNA sequencing, or DDRAT sequencing, which is a next generation sequencing approach that allows to score SNPs across the genome at a reasonable cost as compared to whole genome sequence, for example. Once I obtain the SNPs and score the SNPs, I use then population genomics to identify the different population of mosquitoes and identify the links between the different genetic groups. Finally, to be able to identify the possible introduction routes and invasion routes. So here I would like to open a short parenthesis, because when I started my PhD, I didn't have much experience on population genomics or genetics in general. And through the AIM-COST action, I was granted a short-term scientific mission, and that was an incredible good experience for me. I spent 2 months at the Yale University, which is where they are the top expertise in this topic. So it was in the Adalgisa Caccone lab. And I could really profit of the daily training and of expertise in the group. And I was able to finalize then this project and in general also my PhD. So here I just reported a little video that I did about this experience, and I encourage you to have a look at it or to approach me if you have any question. I'm happy to talk about this great experience. And I go already to the result. So in this map, you see where I collected, or better received, samples from. So from different sites in the invasive and in the native range of the mosquito. And I collected, or I scored 4,700 genome-wide SNPs. And you can see here that I identify in the global sampling 4 different genetic clusters. Which are depicted in the different color. So we have in the native range, Japan, China, and Indonesia. Meanwhile, in the invaded range, US and Brazil. And then we have this big square in Europe. So what's going on in Europe? So if we zoom, so first of all, actually what we are looking at, this is an admixture analysis, which is a program which is used to estimate ancestry. So each line, it's one individual, and the color, it means if it belongs to one or another genetic cluster. And the same is then represented as a pie chart in panel B and panel C. So you see in the native range quite distinct 3 different clusters, and then in the invasive range there are at least 3 genetic clusters, but 2 are domaining. And if you look at Italy or Switzerland, for example, it was my population of reference because it's really in the heart of Europe, it's quite a mess. And so you have a lot of bars with 2 different colors. So what does it mean? It means that there is a high level of genetic admixture and a low level of genetic structure. That means a high level of gene flow, what we will expect. So these regions are very connected. And this can be easily explained by the, no, back, sorry. Okay. So the genetic makeup, the result of this study, can be easily explained by passive dispersal through human transportation network. And this is also backed up by surveillance data. So in this map, I show the surveillance data from Switzerland from 2013 till 2018, where we surveilled the mosquito along the traffic routes. And these are data based on ovitraps. And we can see, looking at the frequency, that there is really a south-north distribution pathway, which is following what is the main national motorway, the A2, which connects the south with the northern Europe. This is also a publication from that we publish in PLOS NTD. So, as you're most familiar with, Aedes albopictus is laying eggs in the spring, which are then surviving the winter, and then hatching in the next spring. So this was autumn. And this process is called overwintering. And very interestingly, in this study, we could find an indirect proof of overwintering based on genetics. So In France, in Strasbourg, we found a couple of mosquito siblings. So 2 mosquitoes coming from the same mother, but across 2 different years. So in 2017, we sampled as eggs, and the year after, we found an adult. And this is an indirect proof that the same female lays some eggs which overwintered over the winter. So, and here I just report the value used to identify full siblings. And this is quite interesting because it's an indirect proof based on genetics. So if we have to summarize the result of this study and play a bit the border control, if we have as a reference the Swiss population, these have been invaded multiple times from Italian sources. But looking in general at Europe, we have a really interconnected mosquito population. So a lot of exchange between also Germany and France. And this is explained by the passive dispersal of a mosquito. If we look at the population in Italy, we could also tackle the introduction pathway into Italy from Chinese, Japanese, and US sources. And this has been already suggested by other studies. So to leave you with a take-home message, the finding of the overwintering population north of the Alps based on genetic data, and in general, the expansion pattern and the genetic background of the mosquito population present in Europe with high admixture point to an increased risk of further northward expansion of this mosquito and call for an increased surveillance and mosquito population control in Northern Europe. And with this, there are many people I would like to acknowledge because many people, many sitting in the room, many connected online probably today, made this project happen. And they contribute with mosquito samples from all over the world. And I could not even fitting them in only one slide. I had to put 2 acknowledgement slides. So again, many thanks to all of you. And many thanks to my collaborators and the funding of this project. And of course, you for your attention. Thank you.